presentation. So you've all seen this slide probably already a couple of times in the last few sessions. So this is a model of the processes leading to to immune memory, adaptive immune memory. We start with a pool of naive immune cells with varied specificity, and the cell that is specific for our invading pathogen gets expanded to generate the acute response and clear the body from the infection. After that, the bulk of these cells die, and a few of them remain as long-lived memory cells, which rapidly respond again upon infection. Recently, there has been some evidence that the innate immune system might be able to develop some kind of memory as well. That's why we're all here today. And two scientists and their team contributed in particular to this idea, and I want to briefly present you with their major findings. This is Peter Abi, a Danish physician who went to Guinea-Bissau in the 80s to fight the incredibly high child mortality there. At that time, many infections were spreading, in particular measles, and no one was vaccinating. Peter introduced the measles vaccine and started measuring the mortality of the kids to gauge his success. And I'm sure Christine will tell you a little bit more about this also in 2 weeks. But I just give you the gist of it. So he soon saw that he was more successful than anticipated with vaccinating against measles, because child mortality was reduced by much more than what was attributed actually to measles deaths. And then he and the WHO did a meta-analysis of many vaccination trials in children in Africa, and they saw a pattern. That live vaccines such as the measles vaccine and BCG have an added benefit and protect against death beyond the vaccine-related disease. And they termed this heterologous protection. And this plot is from one of these meta-analyses where you can see on the x-axis the vaccine efficacy against death, and the y-axis shows you all the different studies. And you can see for many of these studies, you have a significant vaccine efficacy against death here. Now, the question, of course, was, how does this all work? Are cross-reactive T and B cells responsible for this? But then you would need to have plenty of them to convey such a broad protection against all kinds of deaths. So what else could it be? And about 20 years later, Mir Netea, you've probably all saw him 2 weeks ago, a professor at the Radboud University in Nijmegen, and full disclosure, he's also one of the members of my PhD committee when I was doing my PhD in the Netherlands. And he suggested a possible mechanism of heterologous protection, epigenetic macrophage training. And in their key paper, Mia and his group did a simple mouse experiment. They injected SCID mice either with PBS as a control or with BCG vaccine against tuberculosis. And SCID mice have a severe combined immunodeficiency, so they lack T and B cells. So according to the scheme that I showed you right at the beginning, vaccines should have no effect in these mice, right? However, I should mention that SCID mice do have a functioning innate immune cells, including monocytes and macrophages. So after 14 days, Mia and his team injected both groups of mice, the ones treated with PBS and the ones treated with BCG, with a lethal dose of the fungal pathogen Candida albicans, which is, of course, completely unrelated to the pathogen from tuberculosis. And this is where things got interesting. So when the team measured the survival of the mice, they saw a clear benefit in the BCG-trained cohort. And I apologize for the quality of the slides, but this is like zoomed out from the original publication and didn't provide a higher resolution, unfortunately. But I think you can recognize probably the key findings. So on the x-axis, this plot shows the time after infection with Candida albicans. And on the y-axis, it shows the survival rate of the mice in the different groups. In blue, BCG vaccinated, in black, PBS treated. As you can see in the BCG trained group of SCID mice, there's a 100% survival rate, while the PBS treated mice start dying around 10 days after infection. Since the SCID mice lack T and B cells, Mia and his team deduced that the protective effect must be mediated by activated innate immune cells, such as the monocytes and the macrophages. Intriguingly, further molecular and functional analysis suggested that epigenetic changes in innate immune cells are key mediators of these effects and lead to enhanced cytokine production and activation state in BCG-trained monocytes. So based on these findings and some more in-depth studies, Mia and this group proposed a model for innate immune memory, or trained immunity as they termed it. They suggested that vaccines reprogram the epigenome of monocytes, making them produce more cytokines, leading to protection against non-specific pathogens. Now, why could this be important and interesting? Well, this field is still very young, but some scientists suggested that trained immunity could be used to boost immunity in vulnerable populations with impaired adaptive immune systems, such as infant or the elderly. In addition, during a pandemic situation such as COVID-19, non-specific vaccine effects could mediate a certain degree of protection during the early phase when specific vaccines are still in development. So I don't know how you feel about trained immunity after listening to all these talks and being presented with this data. But when we started working on this a while ago, trained immunity was a really new idea. And in our lab, we had many critical discussions on the topic. So working on vaccines and the innate immune systems, we were naturally really excited about the thought of giving innate immune cells also a memory. However, as good scientists, we felt we needed an approach. We felt that we needed to approach this very novel concept in a very agnostic way without any preconceptions. So on the spectrum of beliefs, we place ourselves amongst really the most agnostic people, depending on whom you ask and what day it was. We were maybe more in the agnostic theist corner or more in the atheist corner. But definitely we thought, how can we look at this whole thing in a way without preconceptions? And what are the most important questions to ask? And then we came up with a catalog of many different questions that we thought are important to answer. And here I'm just showing 3 of the main questions that we started addressing. So first of all, we wondered, are these epigenetic changes restricted to monocytes, or can other cells benefit from the same mechanism? Do all vaccines induce epigenetic changes, or is this a special effect that somehow only BCG can do? And then the last question was, is there a way to control this effect and to induce it deliberately? So far, we're always only looking at this in the context of vaccines, but was this kind of a serendipity, a random discovery after vaccination, and other immune stimuli can do the same? Or is this really something specific to vaccination? And yeah, after making this catalog of questions, we got to work. And most of the research until then was focused on the BCG vaccine and on monocytes. So we aim to conduct a systems vaccinology style assessment, looking at the epigenomic immune cell landscape in a holistic fashion and in different types of vaccines. And we chose influenza vaccines as a model because influenza vaccines are an important and interesting area to study. The vaccine is widely used, has variable efficacy, and with a quickly mutating virus, broad non-specific protection would be quite beneficial in this setting. So first, we analyzed PBMCs from healthy subjects that received the seasonal influenza vaccine TIV. We used a range of bulk and single cell tools to determine the epigenomic immune cell landscape at time points before and after administering TIV. As a first step, we wanted to get a bird's eye view of the epigenomic landscape. We were looking for a technique that would be high throughput and that would allow us to screen many immune cells at several interesting time points. So at Stanford at that time, we got in touch with PJ Utz's and Purvesh Khatri's labs, which just has developed a CyTOF-based assay called EpiTOF that would be able to deliver exactly that. Now, EpiTOF is basically a massively multiplexed intranuclear antibody staining modified histone proteins. And the experimental workflow is very similar to traditional flow cytometry or CyTOF experiments. Now, we start with PBMCs and do a standard antibody staining for the cell surface markers to help identify all the different cell types in our sample. Then we fix and permeabilize the cells for intranuclear staining and come in with a second antibody cocktail that is specific for the histone modifications. Then we measure everything on a CyTOF. So this slide gives you an overview of the markers that we identify with EpiTOF. The experiment is split up into 2 panels that we apply in parallel to all of these samples. Both panels have the same 16 lineage markers shown on the very left, and each panel focuses on a specific set of 19 histone marks shown in the middle and on the right. For example, the panel in the middle focuses on phosphorylation and acetylation marks, while the panel on the right focuses on methylation marks. Together, both panels cover all major classes of histone modifications, and PJ Utz's and Purvesh Khatri's lab really put a lot of work into validating all of these antibodies with mechanistic experiments. All right, so this plot gives you an idea of how the raw data of this epitope looks like. So the plot shows the expression of H3K27 acetylation in a few of the major immune cell subsets. You can see the mean levels and distributions differ quite a bit between different populations. So for example, we have the classical monocytes and myeloid dendritic cells. They are rather high on the acetylation level. And then the T cell subsets are a bit lower, for example. And indeed, the histone modification data alone is actually sufficient to identify all major immune cell subsets using a PCA analysis, as we showed in the original paper. But of course, we're not interested really in the differences between the cell types here, but we rather want to know how vaccine-induced changes happen in all of these cell types. So now we compared the levels of each histone mark in all cell types at day 30 after vaccination relative to day 0. And that's shown in this heat map, which summarizes the main results. So on the x-axis here, you have all the different histone marks that we detected with epitope. And on the y-axis, you'll see all the different immune cell subsets that we identified. The color in the fields now indicates whether the level of a certain histone mark in the indicated cell type changed, blue for a decrease and red for an increase. Now, this heat map, of course, is a lot to take in. So I want to direct your attention to a subset of changes in innate immune cells that were among the most significant ones, actually. As you can see here in this plot, monocytes and dendritic cells showed a synchronized reduction in a number of histone marks, mainly acetylation, with H3K27 acetylation being among the strongest ones. Interestingly, the enzyme PADY4, which catalyzes chromatin citrullination and is associated with inflammation, was also reduced. So what this shows is that in addition to the monocytes, vaccines can also modify the epigenome of dendritic cells. And this is a plot zooming in on the kinetics of histone acetylation in myeloid cells. So on the x-axis, you can see here the time after vaccination. The y-axis indicates the differences in acetylation levels compared to day 0. As you can see, there is a sharp drop in acetylation at day 30 after vaccination, which returns to almost normal levels about 6 months after vaccination at day 118. So in this case, the window of these epigenetic changes appears to lie somewhere between 1 and 6 months. So histone acetylation is an indicator of open and transcriptionally active chromatin. Our data of reduced histone acetylation in monocytes and myeloid dendritic cells hinted towards a more restrictive, inactive chromatin environment. To gain in-depth insights into the genes and molecular pathways targeted by these epigenomic changes, we used another technique called single-cell ATAC-seq. And we've already heard a little bit about ATAC-seq before, so I'm going to keep the explanation really short. But ATAC-seq is an assay that detects open chromatin regions accessible for transcription. And that means these regions are either actively transcribed at the moment of the measurement or kept accessible so that the transcription can swiftly be initiated upon a certain signal. Now, we wanted to see whether there was a systematic reduction in the accessibility of genes from certain pathways that is associated with the reduction in histone acetylation that we observed. Now, our approach was rather simple. We isolated innate immune cells from cryopreserved PBMCs using flow cytometry, and then conducted single-cell ATAC-seq and single-cell RNA-seq on these cells, and worked with computational experts from Nir Yosef's lab to analyze all the data. As a first step, we used dimensionality reduction and clustering techniques to generate the epigenomic and transcriptional immune cell landscape. We identified all major innate immune cell subsets that were expected after our enrichment approach. So we have here the monocytes in green, and then different dendritic cell subsets in blue, the myeloid ones, and also the PDCs here. And then our sorting was not perfect, so we have a little bit of impurity from remaining BT cells and also some progenitor cells. Next, we investigated whether the seasonal influenza vaccine induced lasting epigenetic changes in any of these subsets. Now, this slide summarizes the key changes in chromatin accessibility at the transcription factor level. On the y-axis, you can see the different transcription factors. The x-axis indicates the different cell types. Monocytes, again, are in green here at the top, and dendritic cells are in blue. Now, the color in the heat map, again, indicates whether the chromatin accessibility for a certain transcription factor changed after vaccination, blue being a decrease, red an increase. And strikingly, in monocytes at day 30 after vaccination, genes associated with the AP1 transcription factor complex, which consists of members such as FOS and JUN, are closing, meaning genes associated with AP1 are less accessible and less transcriptionally active after vaccination. The same holds true also for the myeloid dendritic cells. Also, the changes here were a little bit weaker. Now, AP1 is an important regulator of inflammation and monocyte development and can control, for example, the expression of TNF-alpha and other cytokines and chemokines. Given this reduction in AP1 accessibility, we wanted to know whether the observed epigenomic changes would have any functional consequences. And to test this, we stimulated PBMCs at day 0 and day 30 after vaccination with synthetic TLR ligands that mimic viral or bacterial signals and that activate monocytes and dendritic cells. After 24 hours, then we measured cytokine production using a multiplex bead assay. The results are summarized here. This plot shows the time point after vaccination on the x-axis and the cytokine production on the y-axis. As you can see, for TNF-alpha, a key inflammatory cytokine, we saw indeed a significant reduction after vaccination using both bacterial and viral cocktails. So each line here connects the two samples from one donor, and you can see for the majority of the donors, we actually see a reduction. Now, in our analysis, we, of course, I told you we used a multiplex bead assay. So we had additional cytokines tested besides TNF-alpha, in total 61 additional cytokines. For a group of 10 innate cytokines amongst these 61, we saw a similar trend of strongly reduced production after vaccination, indicating that the reduction of AP1 accessibility after flu vaccination leads to a state of refractoriness of innate immune cells. So besides TNF-alpha, we have here MCP3 and MCP1, which are important chemokines. There is IL-1 beta, another important cytokine, and many more. Now, how does it all fit into the current state of knowledge? Well, Miai and his colleagues saw an increase in histone acetylation after vaccination with the tuberculosis vaccine BCG, and an increase in cytokine production as well. So we see a decrease in histone acetylation and a decrease in chromatin accessibility at AP1 loci, and a decrease in cytokine production after flu vaccination. So for lack of a better word, after flu vaccination, we see some kind of untraining or refractoriness. So we were really wondering what could be the reason for these differences. And one thing that comes to mind is that TIV is a comparably weak vaccine that does not stimulate the innate immune system very much. BCG, in contrast, is a living pathogen that activates many different innate pathways. So we thought adding an adjuvant that stimulates strong innate immunity would open up the chromatin and lead to enhanced cytokine production. To test this hypothesis, we analyzed samples from healthy donors that were vaccinated with the pre-pandemic avian influenza vaccine from GSK. Now, this cohort contains two groups. One just with an inactivated vaccine similar to TIV, shown in green here, and the other additionally containing the powerful adjuvant system AS03, shown in orange. Now, AS03 is a squalene-based adjuvant containing alpha-tocopherol that induces strong innate and adaptive immune responses. The vaccine is given at two time points, day 0 and day 21. And we then did all the assays that we already did for the seasonal flu vaccine. In light of time, I will just highlight a few of the key findings today. So first, we determined again whether chromatin regions with specific transcription factor motifs were altered after vaccination. On the y-axis, you can see the different transcription factors. The x-axis indicates certain cell types and the vaccination arm, either H5N1 alone, shown in green, or H5N1 plus AS03 adjuvant in orange here. Now, there's two things that we observed here. Number one, monocytes, shown again in green here, after immunization with AS03 adjuvanted vaccine, show a reduction in AP1 accessibility, similar to the seasonal flu vaccine. Same holds true again for the myeloid dendritic cells, but again, to a weaker extent. However, more interestingly, we observed also a strong increase in the accessibility of IRF and STAT family of transcription factors in monocytes and other cells in the same group, for example, also dendritic cells. Now, STAT and IRF transcription factors are important regulators of antiviral immunity and control, for example, the expression of interferon-stimulated genes. Based on this, we wondered about the functional consequences about this increased IRF accessibility. So to investigate this, we looked at some of the additional functional data that we collected in the study. For example, here's the blood transcriptomics data. This plot shows the time after vaccination on the x-axis and the log 2-fold change in expression of antiviral gene modules on the y-axis. As you can appreciate, the antiviral response induced after the second shot with H5N1 AS03, shown in orange, is stronger than the response induced after the first shot. We see a stronger upregulation of these antiviral genes, about 1.5 times, right? So in contrast to that, H5N1 alone is only inducing a minor antiviral response, and there is no increase after the second shot compared to the first shot. So importantly, this enhanced antiviral response after the boost was directly linked to altered epigenomic state at day 21, just before the booster shot was administered. So we saw for genes that were higher expressed here at day 22 compared to day 1, we saw that the chromatin regions that regulate these genes were already increased in accessibility at day 21 compared to day 0. But we wanted to go one step further and determine whether this enhanced antiviral state would provide a substantial advantage in defending against the virus. So we set up a viral model where we used PBMCs from before and different time points after vaccination and infected them with dengue or Zika virus in vitro. We then incubated the cells for 24 hours and measured the number of viral copies. So on the right side, you can see the results plot. The x-axis shows the time after vaccination at which the PBMCs were collected. So day 0, day 21, day 42. And the y-axis shows them the change in viral titers in the in vitro cultures of these PBMCs relative to the in vitro culture at day 0. Strikingly, for both dengue, shown in red, and Zika, shown in blue, we detected reduced viral titers after vaccination, indicating that the vaccine-induced changes to the chromatin landscape might have a direct impact on antiviral defense. And there's one piece of epidemiological evidence that supports this data. So in 2013, a team of international scientists and researchers from GSK conducted a randomized phase 3 clinical trial in 43,000 elderly, comparing the efficacy of the seasonal flu vaccine TIV administered alone or in combination with the AS03 adjuvant system. So this setup is very similar to the study I just presented to you, only that the authors were looking at the seasonal flu as an antigen rather than the avian influenza. And of course, that they had many more participants than we did in our study. So interestingly, the authors found that the adjuvanted influenza vaccine was effective in reducing all kinds of deaths, similar to the beginning where the measles vaccines reduced the occurrence of all kinds of deaths after in children in Africa. So in fact, the total number of deaths here was reduced by 30% as compared to the influenza vaccine without the adjuvant, as you can see here in the table. So without adjuvant, 88 people died during the study. With the adjuvant, only 63. Since this is a randomized trial, these differences cannot be explained by a more healthy lifestyle in the adjuvanted group, as is often the problem with retrospective epidemiological data. So the AS03 adjuvanted vaccine really seemed to provide broad protection here. Also, enhanced protection against influenza cannot explain these differences, as influenza was only reduced by 12% in the adjuvanted group. So this all death effect is really on top of the vaccine effect against influenza. This data really suggests that AS03 adjuvanted vaccines might have a beneficial effect beyond the antigen-specific immunity it provides against influenza, very much in line with our in vitro analysis. So to sum up this part of the talk, our data suggests that influenza vaccines can reprogram the epigenome of the innate immune system in 2 different ways. Number 1 is characterized by reduced cytokine production and closing of the AP1 loci, maybe to avoid excess inflammation or damage to the host. The second type of imprint is characterized by an opening of the IRF loci and enhanced production of antiviral mediators, likely enhancing antiviral resistance. And as we saw with the ASO3 study, these effects can be superimposed and happen simultaneously. Okay, so let's put this all into context. Based on this data, we can imagine innate immune memory as an analog dial, similar a bit to a thermostat. Certain stimuli turn the dial to the right, leading to the type of traditional trained immunity described by Mia and Thea, with increased production of inflammatory cytokines. Other stimuli turn it to the left, leading to the IRF priming state with reduced inflammation, but increased IRF accessibility and antiviral protection. Now, the big question then is, of course, how is this controlled? And one idea that we investigate in this project is a type of stimulus. So bacterial vaccines such as BCG vaccine could lead to traditional trained immunity, while viral vaccines could induce IRF priming. But how general of a phenomenon is this AP1 and IRF priming in monocytes and myeloid cells? Does IRF priming, for instance, emerge in all kinds of immunological situations, like bridges that can be used to cross any gap, be it an ocean, a valley, or a canyon? Or are IRF high and AP1 low cells only emerging after flu vaccination? So we started looking around and we found a few other cases, actually. One example comes from our recent analysis of the BioNTech COVID-19 mRNA vaccine. So in this project, which was led by Prabhu, Madeline, and Thomas, we conducted a systems biology analysis of the immune response to BNT162b2. Today, I will just highlight a few results from a time course analysis of the single-cell transcriptional response that I helped to conduct in this context. So just to give you a brief overview of this experiment, healthy individuals were vaccinated with BNT162b2 at day 0 and at day 21, which is a standard recommended scheme. And then PBMCs at the indicated time points, day 0, day 1, day 2, day 7, day 21, day 22, day 28, and day 42 were analyzed using CiteSeq. Using the CiteSeq data, we then generated a UMAP with all the different cell types. So you have the B cells at the bottom, T cells and K cells on the left, and then this big group of monocytes here in green and pink and red on the upper right. Next, we wanted to see whether one or many of these identified clusters followed a particular kinetic pattern in respect to vaccination. So are there any vaccine-induced changes to the occurrence of the subsets? And a specific cluster, we named it cluster 8, which is a monocyte cluster shown in red here, really stood out. So this plot shows the kinetics of cluster 8, with each line representing one investigated subject. The x-axis shows the different time points after vaccination. The y-axis shows the fraction of these cluster 8 cells within the pool of monocytes and dendritic cells. And if you look very carefully, you can see that there is a tiny population of cluster 8 cells emerging after prime vaccination here at day 1 or day 2 after vaccination, as shown in the little cutout in the upper left. Their magnitude, however, is strongly increased after the booster vaccination at day 22 and day 28, shown on the right here. So this is an increase about 100-fold. So these C8 cells are massively expanding now. Strikingly, these cells also expressed high levels of antiviral genes, including STAT1, MX1, IRF7, and also OAS genes. Now, these findings kind of got us thinking. This pattern of enhanced innate antiviral responses after the booster vaccination that we see here so clearly on day 22 reminded us very much of the pattern of enhanced antiviral immunity and epigenomic changes that we already observed in the avian influenza vaccine with the AS03 adjuvant. Just as a reminder, after H5N1 AS03 vaccination, the modified monocytes and dendritic cells were characterized by increased IRF levels and decreased AP1 accessibility. Strikingly now, when we go back to the COVID vaccine and we plot the expression levels of IRF and AP1 transcription factors in these cluster 8 cells relative to all the other monocyte and dendritic cell clusters that we identified in this vaccine, we see a similar pattern. Compared to their resting counterparts, C3 and C7 and so on, these cluster 8 monocytes expressed high levels of IRF and STAT transcription factors and low levels of AP1 transcription factors. So whether the C8 phenotype that we saw here on the transcriptional level is also imprinted in the monocyte epigenome during immunization with BNT162b2 is currently under investigation in the lab. Since we identified these cells in the COVID-19 vaccine in adults, we also identified a similar subset of IRF high and AP1 low monocytes in lymphoid tissue of mice. Sorry. Okay, so far we talked a lot about vaccines, but what are vaccines but engineered mimics of natural infections, right? So an obvious question is, do infections also induce epigenetic changes to the immune cell landscape? Especially in our context, we were interested in viral infections. And an excellent opportunity to study this question came actually up when we investigated immunity to SARS-CoV-2 in babies and infants just after birth. So this is a study that we conducted together with Mary Start at the Cincinnati Children's Hospital, who had provided us samples from a very extremely unique and fantastic cohort of pediatric COVID-19 patients. So in this cohort, we had access to matched pre-infection, acute time point, and convalescence PBMC samples from infants and young children that were around half a year or 1 year of age at the time of infection. And children, of course, are really interesting cohort to study in the context of trained immunity because their adaptive immune system is not fully developed yet. So innate immune memory could be really useful in this case, right? So due to time, I will just focus on the epigenetic changes here, but we looked at all aspects of the immune response, which you can all find in the paper if you're interested. So what we did here is we took PBMC samples from a subset of these pediatric COVID-19 patients from before infection, acute and convalescent time points. And then we used single-cell multi-omics to determine the transcriptional and epigenetic immune cell landscape. So here we have the paired single-cell ATAC-seq and RNA-seq data on the same cell type available. The results are shown here. So again, we made a UMAP. We identified all the critical cell types, B cells at the upper left, T cells upper right, and then the monocytes and dendritic cells at the bottom left in green in this time. Importantly then, when we compared how these cells were distributed over the different time points, we observed that some monocytes primarily emerged during acute infection, shown in red here. Right, so there was a cluster of activated monocytes, and these monocytes expressed high levels of interferon-stimulated genes, many of which we already saw in the cluster 8 cells of the COVID-19 vaccination subjects. Looking at the epigenetic landscape, you might have expected this already, but we discovered that these cells that were highly accessible at gene loci of the IRF transcription factor family and low at gene loci of the AP1 transcription factor family. So these cells are again very similar to the cells found after the H5N1A03 vaccination and also the cluster 8 cells after the COVID vaccination. A recently published study observed also similar epigenetic changes in monocytes of adults from COVID-19 patients. Interestingly, the frequency of these IRF high and AP1 low cells in our infant cohort was highly correlated to plasma interferon levels. So the same holds also true for the cluster 8 monocytes after COVID-19 vaccination, suggesting that maybe the plasma cytokine levels might be involved in inducing or maintaining this imprint. Collectively, these data show that infections can induce epigenetic changes in monocytes as well, and thus the concept of innate memory is not limited to vaccines, but also extends to infections and perhaps also other diseases. So we see these AP1 and IRF subsets now popping up in many different situations after influenza vaccination, after COVID infection, also in mice. This is really suggesting that there might be a broader or a more generally applicable mechanism at work here. To explore this in a more systematic way, we are currently analyzing epigenomic changes in a broad range of additional vaccine types, including the live attenuated vaccines, mRNA vaccines, and more adjuvants. This is all in a viral setting, but we're really trying to compare different vaccine modalities and catalog a little bit or see whether we can identify patterns in the way that they induce epigenetic imprints. This work is still ongoing, so I won't be able to go into any details today, but we've collected many additional samples and are currently in the process of comparing unique and shared epigenetic imprints at the TNF level and also the gene level. This slide is just a tiny teaser showing you all the single cells that we are working on. And this is, again, a collaboration also with Nir Youssef's lab, especially Ushir, who is working really hard here. All right, which brings us to the last part of my talk, innate memory. What is it good for? So what are the advantages of innate memory? Well, it reacts fast, it is broadly reactive against a large range of pathogens. So to harness innate immune memory, no knowledge on the specific pathogen is really needed. One situation where this could be helpful is a pandemic with an emerging unknown pathogen. And during the COVID-19 pandemic, science and medicine accomplished really something extremely unique and great, which is designing a vaccine in only 1 year, essentially slashing the previous development time by a factor of 10. And I can imagine many of you probably contributed to this great achievement. However, 1 year without a vaccine is still a pretty long period during a global pandemic, as we all experienced firsthand. So you could imagine that a vaccine that provides broad, unspecific immunity based on innate memory, and that is off the shelf available on day one of a pandemic, could be really useful for reducing casualties and burdens on the hospitals. Another area where innate memory could be useful is as an add-on on conventional vaccines, right? So many vaccines take about 1 month or 2 months to generate adaptive immune protections. Sometimes 1 shot is enough, sometimes you need maybe 3. And many of you who have traveled know that you have to start planning your vaccination early to be protected at the start of your trip. Now, adding rapid and broad resistance against a pathogen, against a range of pathogens via innate memory could really be helpful in these situations to bridge the early phase when the adaptive immune system is still learning. And finally, of course, there are many individuals that are not enjoying the magnificent services of an adaptive immune system. For example, infants and elderly are known to have less efficient T and B cell responses compared to young adults. In addition, there's also a large group of patients with a variety of diseases that lead to compromised adaptive responses, such as transplantations, cancer, autoimmunity, or inborn errors of immunity. So innate memory could help protect those individuals. Yeah, moving forward, there are a few key questions that are important to be addressed. Can innate memory be effectively employed to protect from infection or severe disease? During the COVID-19 pandemic, there were a couple of clinical trials that looked at the effect of, for example, BCG vaccination on protection against COVID-19. And I think there were mixed results there. Some studies found some effect against airway infections, but other studies found no effect in other settings. So this is still a very much open question that needs to be investigated. Does this provide any clinical benefit? The other question is, are currently available vaccines really the best way to induce innate memory, right? So I told you already earlier, this was kind of discovered as a serendipity effect when people investigated the effects of vaccines and saw, there's something happening also in the innate memory. But is this really the best way we can train the innate immune system? Or are there other ways, for example, through adjuvants or through the stem cell transfer, as we saw in the talk earlier today? And then finally, who is the target group of that innate memory-based vaccine? And this goes a little bit together with the first point, whether it's effective. And perhaps it's not effective for everyone. Perhaps if you're healthy and in the middle age range and you have good working T and B cells and already have received a lot of vaccinations in your life, you're well protected against many diseases. But perhaps, and this innate memory doesn't do much for you, but perhaps if you have a compromised immune system, and your T and B cells are not working so well anymore, this innate memory could actually help you protect you from infectious diseases. So now in my lab in Tübingen, we're starting to look into some of these questions, and we've received generous funding from the German Research Foundation to investigate vaccine responses in cancer patients. And we've conducted a clinical trial where we collected blood samples from healthy donors and cancer patients before and after vaccination. And as part of this analysis, we are now analyzing in my lab whether there are also epigenetic changes happening here, either induced from the cancer or from the vaccine. And we want to find out whether vaccines can still induce these epigenetic changes that we observed in the healthy people in a suppressed, compromised immune system of cancer patients, and whether this could actually benefit their defense against pathogens. Yeah, hopefully next time I can tell you more about this all. And now what's left for me to do is thank all the great people at Stanford and beyond and in Tübingen who made this work possible. Yeah, just want to highlight a few key people. Chun Feng, who helped with all the viral infection data. Zanne, who helped with the ATAC-seq experiments. PJ Utz and Purvesh Khatri's labs, of course, who helped with all the epitope data. Nir Yosef's lab, especially Tal Ashurach and Rushir Rastogi, who helped with all the single-cell ATAC-seq analysis. And particularly, of course, I want to thank Bali for all his guidance and support during my time at Stanford, and also now that I'm starting as an independent research group leader. And yeah, I want to thank you for your attention, and Anne-Sophie and Jean-Daniel for inviting me to the Vaccine Club.
Thanks a lot, Florian. So you got a lot of congratulations and thank you on the chat for your presentation, and many questions too. So let me ask you some of them. One question is from Jean-Daniel regarding the vaccine schedule. And regarding the fact that you might need to increase the time interval between the prime and the boost to get a good antibody response, to let the time to the immune system to mount this antibody response. And so this time between two injections is an important factor for the GC reaction. And maybe increasing this time between the prime and boost will also influence the epigenetic changes that you describe. So what do you think about this dynamic kinetic issue?
Yeah, I think this is a pretty good question. So I think there are many different factors that play into this here, right? So obviously the dynamics are important, and we're talking about innate memory, but of course they're not, the monocytes and dendritic cells are not living isolated from all the rest of the immune system, right? So I think the way that I imagine this is like the B cell response, the T cell response, the monocyte response, they're all influencing each other. And probably this effects that we see on the monocytes, this imprinting of IRF, AP1, and so on, they might also be driven by cytokines that are produced by other cells like T cells and B cells, right? For example, I showed this correlation with the plasma interferon levels, right? So in this case, it was interferon alpha, but in the COVID vaccine case, it was interferon gamma, and this interferon gamma is probably produced by T cells. So my response to that would be, we don't know at this point, but I really believe that these different kinetics will have an impact on the type of the innate memory and also perhaps on the duration of the innate immune memory, just due to all these interactions between the different arms of the immune system.
Yeah, and actually, Jean-Daniel had another question regarding your study with babies and kids regarding the epigenetic changes. Did you have a chance to compare kids or neonates with adults, and whether the changes are similar in terms of magnitude and quality?
Yeah, unfortunately, in this case, we didn't have a chance to compare directly in the same study the imprint in adults with the imprint in infants. I mentioned there was this other paper from Chong in Cell in 2023, which looked at adults only. So if you put both of these studies next to each other, you can kind of get the non-perfect comparison, I think. They saw very similar changes, I think, to what we saw here. However, there was one big difference, which is that in our case, the duration of the epigenetic imprints was rather short. So when we looked at the convalescent samples, about 1 to 12 months after the infection, we saw only traces of AP1 and we didn't see much of the IRF imprint anymore. And this might have to do with the special situation that children are in, right? So they're, on one hand, their immune system is still maturing. That might bring epigenetic changes with itself, right? But they're also exposed to much more pathogens and infections than we are, right? So we had data every week, twice a week, actually, the kids were sending in their nose swabs and they were analyzed for a broad panel of viral and bacterial infections. And actually, you could see that every 2-3 weeks, they had every kid has infection with a lot of things. I think everyone who is a parent here will probably know how often these kids actually get sick. And then they also receive a lot of vaccines, right, in the first couple of years. So this will all have some kind of imprint on the epigenome, which might kind of muddle a little bit the COVID-19 infection imprint and make this a bit more complex than in the adult case.
No, you're right. Then Christine Ben, who was here, also had a question or maybe a fear regarding your data about the non-adjuvanted inactivated vaccine, whether it could lead to increased susceptibility to infections. And she also mentioned in her text some data from Guinea-Bissau using a H1N1 campaign that was associated with increased overall mortality and especially in females. So what are your recommendations? Do we need to worry? And can you study this in your cancer patients too?
Yeah, so I don't really know of, we didn't test this. I know about the epidemiological studies, which of course come always with their certain limitations. So the way that we see it is that this is a refractoriness that is kind of protecting the host from pathology due to the infection or to the vaccination. So while these pro-inflammatory cytokines are reduced, other parts of the immune system might be still providing the protection that you need, but you would have less of this inflammation that maybe also caused most of the severe disease pathology, for example, in COVID-19. And what was the second part of the question?
So it was about if you can measure this within your new study involving cancer patients.
Right. So the studies that we do, they're usually aimed at mechanistic investigation of the immune system, right? So they're too small to look really at any clinical effects. That's why we have to rely on other bigger studies to get insights into that. So also the cancer patient study will be too small to look into this. But interestingly, there are some studies coming out from cancer patients that were treated with checkpoint inhibitors and that received the influenza vaccine as part of their standard care. There's a larger study from Italy that was released last year, and they had about 50% of the patients accepted the influenza vaccine, 50% didn't want to have the vaccine. And then they compared basically between these two patients, the overall survival on this treatment. And they actually saw that patients that accepted the influenza vaccine had about a 7 months, I think, overall survival compared to the patients, exactly same therapy, same type of cancer, same age, same sex, and so on, but just didn't want to have the influenza vaccine, and they lived 7 months shorter in that study. So it might be that also in the setting, actually, together with maybe some effects of the tumor, this refractoriness could be beneficial.
Okay, great. Anyway, Dr. Christine Ben will be one of our speakers for the next webinar, so we will know more about this kind of study. You have another question by Marie Murat, and actually I had almost the same question. Is the adjuvant alone sufficient, or do we need also still an antigen? And what is the crosstalk between the antibodies or the specific T cells, and is the adjuvant alone enough?
Yeah, so I would love to do this kind of experiment, but we were wondering in a human setting, that's really tricky because usually you don't have a situation where you can just apply the adjuvant, right? So we'll investigate vaccines that are licensed and there's always an antigen attached to it. So the best we can do is basically what we're trying now, where we compare different types of vaccines that all have a different antigen, also have slightly different adjuvants, but you can perhaps, or that's our goal at least, to draw some conclusions from that, where you can see, okay, independent of the antigen, the adjuvant does always induce this type of pattern, right? Or perhaps the response is, the result is later the other way around, that only this adjuvant does something, but this adjuvant does nothing, right? So that's kind of what we are investigating right now in this bigger comparison of the vaccines. My hypothesis again would be that it probably depends on both, because I believe also that the cytokines produced by the B cells and the T cells will have an impact on innate memory, and in this case, you will need an adjuvant an antigen to also stimulate the T cells to produce their interferon gamma, for example, or other cytokines, right? So I think the main effect might be from one or the other, but probably the result we observe will always be like a combined effect of all the different immune arms.
You also have a question from my colleague Nabila Siddiqui, who is interested in pediatric immunology and infectiology, regarding your study with the infants. Were you able to see any subgroups in terms of epigenetic marks? And can you tell us more about the post or long COVID in your study group? And is there a link between the epigenetic marks induced or resulting from the infection and the long-term effect of this infection?
Yeah, that's really exciting question also that we were wondering ourselves. Does COVID infection basically leave some longer imprint that has an effect on how the immune system develops, and whether it can cause some of these long-term effects? Unfortunately, in our cohort, we had too little subjects also to investigate this. So we had here 10 infants that we investigated, and I think to answer such a question, you need much larger cohorts really to be able to stratify them in subset. Right, so all the infants were also not exactly the same age, right? So there's some variation always that also affects these responses. So then you're having too many factors to further subdivide the cohort, unfortunately.
Thanks. I had myself one question. So, of course, by studying PBMCs, you can analyze monocytes and disease. Did you have the chance to look at neutrophils or other myeloid cell populations that are also important in the innate defense?
Yeah, right. That's definitely an exciting topic. So in these studies, we were working with frozen PBMCs, unfortunately, so there were not really any neutrophils left. But for example, in the study with the cancer patient that we started now, we also have fixed whole blood. So there is a large population of granulocytes. And there with flow cytometry, we also want to investigate this a little bit, yeah.
Okay, great. Camille Locht from Lille asked you a question about the microbiome that likely has an impact on the ability to induce one or the other shift in the epigenome. Is it something that you are also looking at, or what do you think?
Yes, we actually looked at that in the first paper that I was talking about, the comparison of the flu vaccines. So I didn't mention this here because I thought it was maybe making the talk a little bit too long. But in this first study with the seasonal influenza vaccine, we actually had two groups of subjects, one that received antibiotics before and during the time of vaccination, and the other group that just received influenza vaccination. And the antibiotics were really wiping out the microbiota in these patients completely in the gut. so that at the time of vaccination, basically, there was not much left there. And so we compared before, so at baseline, at day 0, before the vaccination, the epigenomes between the antibiotics treated and untreated patients, and we actually didn't see too many differences. We saw some traces of epigenetic changes that became visible in ataxi that we couldn't see really with the epitope. And those were in line with kind of the data that was reported in the original study. So in the lab, there was a study before that looked specifically at the influenza-specific antibody response upon this antibiotics treatment. And we just piggyback basically on their samples. So they saw that there's more inflammation in the case of the wiped out microbiome. And we saw traces of this inflammation at the baseline as well, but they were not really strong. And we were also really surprised by this because that was one of the hypotheses going to this project that there might be an effect. Yeah. And then based on the vaccine effect, the TIV-induced effect, that was really exactly the same in both groups.